Hello people of the world. I'm going to talk about mermaids today for Nat 5 biology. Just kidding. Again, just my daughter's mermaid. I like them, they're colourful. Been a bit out of it with pneumonia the last few weeks, but I'm slowly getting well again. Okay, so enzymes, they have optimum pHs, okay, plus temperatures. Normally measured in degrees centigrade. You don't usually work in Fahrenheit, and normally Kelvin. Okay, just for reference check, 273 Kelvin equals 0 degrees centigrade, okay? So at 25 degrees, 298 Kelvin equals 25 degrees centigrade, okay? The reason it's 2730 is because so 0 degrees Kelvin equals minus 273 degrees centigrade, which is absolute zero. Okay, so this is normally when you're in chemistry labs, biochemistry labs, when you're working in scientific labs, you actually work in Kelvin, okay? You work in Kelvin, not centigrade and Fahrenheit. So 273 degrees Kelvin is 0 degrees centigrade and so on. So yeah, for the record, it's impossible to actually get to minus 273, but this is the coldest possible temperature that can exist in theory, although you cannot actually get there. I think science has got down probably a few millionths of a degree above it, but it's impossible to actually get to it. Anyway, so enzymes. So we got the optimum pHs, optimum temperatures. So I showed you before how it was. You have an enzyme, say, okay, and that's the active site, okay, and a substrate comes in, okay, and then the substrate binds, so you recreate that structure. Okay, that's the substrate, okay? And then so you get a substrate enzyme complex. So you get, this is the, I'll just call it the substrate enzyme complex, okay? So in this state, the substrate, whether it's glucose, it could be glucose, it could be fats, proteins, etc. The substrate is broken down, is changed, okay? So the substrate, which is or normally in digestion in your body, it's broken down. But in other chemistry in your body in, say, respiration, when an ATP is made, okay, during growth, in that case, the substrate undergoes a change and becomes a product, okay? In this case, it becomes a product as well. The product's broken down. The product's the broken down part version of that. That's broken down. But you can have a product, okay? So in normal enzyme reactions, so substrate is converted to product, okay? In various parts of sort of different biochemical processes in the body. Okay, so yeah, temperature, in order to make this process occur optimally, you need the correct temperature. In the human body it's 98 degrees centigrade approximately. In plants it's cooler temperatures because they're outdoors, and in say very northern countries like Norway, Sweden, Canada, the temperatures for enzymes to work in plants are much cooler temperatures. In tropical countries, where it's very hot, the optimum temperatures are much hotter. So that's why, yeah. And also, you got the effect of, I mentioned pH. Okay, well, in most life on Earth. Pen's going to dry. In most life on Earth, pH is 7. Okay, that's the pH of water, and water's what makes life tick. So, so after the so substrate enters the enzyme. Once that's finished. So you got the substrate goes into the enzyme, okay? Okay? And then this changes, so you get okay? This is very diagrammatic, but it changes, and then excuse me, and then you get substrate leaving as so that's the product. So in this case, the substrate is broken into 2 bits. Okay, that's the product. So yeah, so this is how enzymes work. Substrate attached to enzyme, you get a substrate enzyme complex formed, enzyme substrate complex. A reaction occurs, or you get the substrate, whether it's glucose or fats or proteins being broken down, or plant material being broken down, meat being broken down, sweeties being broken down, and then you get the substrate is released as a product, and the enzyme, it returns to its original form, okay? Must emphasize, enzymes, catalysts, they're not changed during a reaction. The final state is equal to their initial state, okay? They're not changed. All they do is convert a substrate into a product. The enzyme might undergo a bit of rearrangement during the reaction, but by the end of it, it's exactly the same as it was at the beginning, okay? An enzyme is not changed in the reaction. Okay? Is that everything on the subject of enzymes? I think so. I could show you a quick sketch of, whoops, on camera. So you have basic, if you plot temperature, okay, increasing enzyme activity, so that's increasing enzyme okay. So a typical for temperature, you might have sort of 0, maybe 10, 20, 30, 40, 50, okay? So you might have in mammals as both humans, you have the optimum temperature around about 38 degrees, okay, roughly, okay? And so the activity, the enzyme will be really low at the start, okay? At 0 it's basically 0, okay? And then it gradually rises, okay? Until it reaches an optimum at 38 degrees centigrade roughly. And then when you get above that, it starts falling again, okay? But actually when you go up to high temperatures, it falls off pretty fast. When you go up to sort of 78 to 1900, it really goes down to 0, okay? So at 0 degrees it's 0, and minus 0 is 0. It increases to the optimum and then falls again. pH graphs are actually rather similar to that. So the pH graph, pH versus enzyme activity, is actually kind of similar. At low pH, whatever the optimum pH is, below that it's low, it gradually rises to an optimum and it falls again above the pH. Okay. Optimum pH, okay? I mentioned pH is similar to this. Quickly show you the pH graph. So you got so pH 0, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, okay? And you got increasing, so with enzymes you got increasing rate of reaction. That's increasing rate of reaction, okay? Is that right? Okay, so that's reaction. So something like let me think. So okay, that's pepsin, okay, which is a stomach enzyme that breaks proteins down. Okay, so that is an optimum pH of about 2.5. Below that and above that, the activity falls off. But at 2.5 it reaches a peak. So next one is about pH is 7, neutral. So you got round about okay, so this is most enzyme. Most enzymes, so pH optimum 7, okay, I should have drawn the peak actually on that line, but slightly, okay. So below that, below 7, it's that rate of reaction is low, it gradually increases at 7, it reaches a peak and then it drops off, okay? And then alkaline, and then pH 10. So you got, again, optimum that, and optimum that, okay? So this one is called alkaline phosphatase, okay? Alkaline phosphatase, if I can write this all in, it would be help. Right here. Alkaline phosphatase. Okay, so that's alkaline phosphatase, an enzyme. I forget what it does, but has a pH, optimum pH at pH 10. Below that, that activity is lower, and above that activity is lower. Most enzymes are pH 7, and pepsin, the same with trypsin, the stomach enzyme. These digest protein. They break peptide bonds, optimum pH at 2.5. So below that and above that it's low. So that's, again, a similar kind of shape of graph to this, to the temperature graph, okay? So this is the optimum pH graphs. Yeah, that's role in bone formation, okay? There's a role in bone formation. Okay, that's